Okay, hello everyone. So for this video, I'm going to show you how to run the case of the dirty dirt. It's a soil test. Please pause the video and kind of take a quick read through the scenario as to what you're looking for here with the missing person and then how they are found a year later. We're going to be looking at how we can match up where this soil may have come from, see if we can get any possible suspects out of it. Okay. So what we have for our lab is we have our filtering cylinders. I already put cotton in the bottom. It has a little hole in the bottom as well. And that's going to allow the water to run through that soil sample. We have our distilled water. We also have our arsenic test reagent. And we have our lead test reagent. Okay. And then we have our 3 soil samples from our suspects, suspect 1, suspect 2, and suspect 3. And on your answer sheet, they have noted where they have been from, right? Suspect 1, we took the dirt from the backyard garden. Suspect 2 was a dirt pile in the side yard. And suspect 3 was from the front yard flower bed. Trying to see if anyone matches up. So here we go with running this lab. The first thing we're going to do is take 2 of our cylinders. Oh, and we also have medicine cups to catch the water in, right? So I'm going to put a scoop of suspect 1. I'm going to send out of the picture here. I'm going to run this. We're going to put a scoop of suspect 1's soil in here. Okay, maybe a little bit more. And we're going to put a scoop of suspect 1 in the second vial because one test is going to be for arsenic and the other one is going to be for lead. All right. Then what we're going to do is take our medicine cups. Remember, this is suspect 1's soil sample. So I'm going to put 15 to 20 milliliters of water through each test tube. So here's one. We're going to pour it in rather slowly and let it run through. You can kind of see it's going to run its way through. I'm going to do the other one simultaneously. And the idea here is the cotton is supposed to stop the majority of the soil from running through. Okay, so as you can see there, fairly clear. Nothing wrong with that. The second one. Same thing. I shouldn't say the second one. The second column, which is still suspect 1. So we have 2 cups, suspect 1 soil samples run through. First thing we'll do is the arsenic test on the first cup. So here's our arsenic test. And if we get a blue color, it means that it is positive for arsenic. So if I put this in, 1, 2, 3, 4. You can see that we stayed orange with that test. So that means we would be negative for arsenic for suspect 1. And then if we take the lead test, run that. 1, 2, 3, 4. And that told us in the directions that if we got a pink color, it would be positive for lead. And we don't have a pink color. It's got a little bit of an orange color just because of the soil. Okay, so for that's suspect 1's results. You should be marking that in your data chart as we go through. Now we'll move on to suspect 2. Suspect 2, we're going to do the same thing. Once again, clean vials, these filtering chambers. Suspect 2. A scoop of dirt in each one. Okay, we'll now get our medicine cups them inside of there so they can drain through. And we're going to do the same thing. Take our 15 to 20 mLs of water. I'm doing 15 for today. We should try to keep it consistent throughout your test. So we'll do 15 with all of these. Here's 15 for the other vial. Remember, this is suspect 2. It ran a little bit dirtier. Should have maybe put a little bit more cotton in the bottom, but that's okay. And that has just about completely filtered through. So put that aside. Here's the other one. I want to put a lot of cotton in the bottom, so taking a little bit longer to run through, but that's okay. Okay, suspect 2. First test we'll do is the arsenic test. Let's see if we can get a color change here. Okay. Definitely a color change to blue. That was the arsenic test. And if we do the lead test, this one, once again, will turn a pink. It just stayed kind of orange, just like the soil. So that had no reaction. Okay, your arsenic test turned blue, and your lead test had no reaction. Move on to suspect number 3. Same thing, 1 scoop. And another scoop. The second vial. I missed a little bit there. Okay, suspect 3. Let's get 2 new medicine cups. Notice that we want to use new medicine cups just because we don't want cross-contamination. mLs going into the first one. mLs into the second one. And what I'll do is I'll hold these up so it allows it to drain so it doesn't get plugged up on the bottom. You can see that it's running through. It's a little murky, not a problem. Okay, this is suspect 3 once again. A little shake off. Okay, so our first test we're going to do is our arsenic test. Arsenic will turn blue in the presence of, or this arsenic test will turn blue if there is arsenic found in the soil. Four drops. See for yourself the color change. And our lead test. Sorry, I didn't let you guys see that, but there is no change there either. This just stays that darker brown. We would get a pinker color, pink fuchsia color if there was lead in the soil. Okay, so that was suspect 3. Here was your arsenic test. There was your lead test, which didn't show any color change. Okay. So as you're working through this, you should have been filling out your chart. Okay. You do have the ammonia test that has been completed for you already. We did not run that in the lab, but I gave you the information. Then you're going to break down your chart and answer your questions and submit that by the end of the period. Okay. You will need that opening paragraph or the opening reading to answer just a few of those questions, come to an analysis and a conclusion on a few of those questions. Okay. If you have any questions while viewing the video, by all means, talk to me through Zoom. But it is a fairly straightforward lab, and hopefully we don't have any problems.